Hello and welcome to the Research Reproducibility Conference, Educating for Reproducibility Research Integrity. We're really happy to have you here today. My name is Melissa Ruthlefson, and I am the chair of this conference, as well as the Associate Dean and Faculty Director at the Health Science Center Libraries at the University of Florida. Though COVID obviously has been a terrible catastrophe globally, one of the small benefits of having COVID happen is that our original conference, which was scheduled to meet in person in March, was canceled and delayed until today. And that has enabled us to get attendees from 21 different countries, 39 states and Washington, D.C., as well as from 181 different institutions and companies across the world. So welcome to all of you. We're so happy to have you here. And we are going to be starting momentarily, but first I'm going to give you just a few pieces of information about using the system. Many of you have already logged in and joined a table. We're of course using this remote platform, and we will be encouraging that you turn on your video and or your microphone during our breaks and discussion sessions and the poster sessions as well, because you can actually talk with your colleagues at some of these small tables and hopefully meet some new people and get into some great conversations. You can also chat with the other people at your table, or you can use the general chat that is available in this presentation mode. You can also hide that if you wish, but we will be requesting that any kinds of questions that you have for our speakers, that you use the Q&A feature that is also linked on the right-hand side. We will be monitoring the chat for things like technical issues. So if you have problems accessing the platform, or if you are struggling to hear or have other technical issues, please put those in the chat. But any kinds of questions that you have for any of our speakers, paper presenters, and so on, please do put those in the Q&A so that the moderators will see them. We will be recording all of our main sessions as well, and we will send out all of the links to all of the people who have registered after we have those available. It will take us a few weeks of post-production to get those available, but they will be available for then your use as well. One of the questions that we've already gotten is, will the presentations be available on after the conference? And we will be requesting that our paper and poster presenters, as well as our other speakers and panelists who are willing to do so, can share them on our OSF conference site. And again, we'll be sending that information out to everyone who has registered. One thing also to mention is that we have set up a great system of cooperative note-taking if you're interested in using in Google Docs. There is a link to that on the bottom of the main screen. Otherwise, we will also be sharing the link to that the Google Docs notes page throughout the session. There will be two separate documents, or three actually, one that you'll go to that will have the agenda, and then you can link out directly to where the notes for that particular item are in the other collaborative note-taking documents. Another thing that we're going to be doing that's a little bit unique is we really do want to have this be not just something where you sit and listen, but something where you participate and contribute to our discussions. So tomorrow we're going to have an unconference session or breakout discussions, and we would love for you to submit your topic ideas on what you would actually like to discuss. And we'll collate the top themes for those discussions, and then we will label each of the floors within the remote platform so that people can go to whichever floor they would like to have a discussion on that topic. If you want to submit a topic that you would like to see discussed, we'll put in the chat a link to the Slido app that we're using. We're going to be using hashtag reproducibility2020 for using Slido to submit the unconference topics, as well as if you are on Twitter and want to tweet anything today, we are using that hashtag reproducibility2020. It's also linked in the title page of the conference. A few other things to mention during some of our sessions where we will be back at the tables. There are 2 lounges that are available from companies who had requested to participate. EBSCO is here to talk about protocols.io and CodeOcean, if any of you are interested in talking about either of those with their representatives. And Lab Archives is also here in one of the lounge sessions. You'll see all of the tables and the lounges have labels underneath, and those will give you indications of which table that you're at. And that will be important later today for the poster session because the different posters will be at certain tables as they are linked and indicated. So those are just a few of the Q&A. Again, let me welcome all of you here. We will be available to help you throughout the session, and we do again encourage your participation throughout the day. I'm really looking forward to it. So now I'm going to turn it over to Dr. David Norton, who's the Vice President of Research here at the University of Florida, and he will say a brief few words for you. Dr. Norton?
Good morning, good afternoon, good evening, depending upon where you're joining the conference. Let me, on behalf of the University of Florida, welcome each of you to the Research Reproducibility 2020 conference. We very much do wish that we could all be here in Gainesville, Florida. If you were here, you would be experiencing, I think, the coldest day of 2020. It was 28 degrees Fahrenheit this morning, which for Floridians is brutally cold. For those in other parts of the world, it's not so much. But in any case, wherever you're at, welcome to our conference. Let me first start by just congratulating the organizers of this conference in the challenging times that we're in, when things have been planned and we're thrown this curveball called COVID-19. It presents a number of challenges, and the organizers for this conference did an amazing job of pivoting, looking at the various technology and platforms. I just want to congratulate them on putting together really an outstanding program. The University of Florida is a very research-intensive, large institution, over 50,000 students. But from my perspective, the thing I worry about is the research. We do over $900 million per year in sponsored research. One of the most important components of that is the integrity of the research that we do. If we lose that integrity, regardless of the quality of what we do, we lose our effectiveness. And reproducibility, the integrity of what we do, disclosing who is funding our research, how those researchers are influenced is so important. We have a team that works on that, educating the faculty, and I'm sure that those participating from the University of Florida will learn quite a bit from this conference, but each of you, it's really important. We all live in a time where clearly research is bringing us a solution to the COVID-19 epidemic. The integrity of the research behind those treatments, those vaccines, is obviously important. And so it couldn't be more visible as to how important this topic is. So again, I just welcome you to the conference. I wish you great success going forward. And with that, I am going to turn it back over to the organizers, to Melissa.
Thank you so much for being here, Dr. Norton, and thank you for that welcome. Certainly, we are very much convinced that reproducibility and the related concepts of transparency, rigor, replicability are all really key for our learners to discover so that when they become the part of the research enterprise, that they have the skills and the knowledge to really embrace open science and transparency and make sure that the research that is being done is done with integrity and is rigorous. So thank you again so much for being here with us today. We're going to start our keynote in just a few minutes. But I do want to, again, remind you that you can check in the chat for links to our Google Doc that will allow you to do collaborative note-taking, as well as the link to the Slido app where you can submit topics for the unconference sessions that we'll have tomorrow, the breakout sessions. And that will be tomorrow afternoon. And we will be soliciting all of your notes and comments and feedback for that throughout the duration of the conference. So feel free to jump in and submit your ideas and the things that you want to talk about at any point. We are going to start introducing Dr. Nosek in just a few minutes. I'm not seeing any questions come through at the moment, but do feel free to use the chat to ask any questions, or if you have questions for the speakers or moderators, please, again, use that Q&A panel. You will notice also in the agenda that we have sometimes concurrent sessions. We're currently in Building 1, which will be where paper session 1 and other Building 1 topics are. We will let you know when it is time to choose between Building 1 and Building 2, which will have the other concurrent sessions. So we have 2 concurrent paper sessions, and we also have concurrent lightning talks later today. We'll also pause between each of the paper presentations. So if you want to jump between buildings, that will be an option for you. And we'll just keep those notes coming as in the different announcements that are being sent team, as well as in the chat for any kinds of instructions that you might need. So we are here to help, and we will also mention that we are putting links to the entire agenda if you want to plan out what buildings that you intend to go to later in the afternoon. You can plan that out in advance, but again, we'll kind of prompt everyone through that as well. Okay, so I think seeing no additional questions come through, we will go ahead and get started. Welcome to day 2 of the Research Reproducibility 2020 conference, Educating for Reproducibility Pathways to Reproducibility. My name is Melissa Rathlifson, and I am the chair of this conference, as well as an associate dean at the Health Science Center Libraries at the University of Florida. And we would like to, again, extend a welcome to all of you who are able to join us here today. We have a great set of sessions planned for you, and we're really excited that we have such wonderful speakers coming up, and we hope that you enjoy as much as I know that I'm going to. Just as a few reminders, for those of you who haven't been here yesterday or need a bit of a refresher, we do have a link in the bottom of the screen to our Google Doc where you can link off to take notes on specific events that you actually attended. In addition, we would like to remind you that you can submit topics for our breakout discussions or unconference session using our Slido app, and we will put the links to both of those in the chat. And so Hannah, thank you for just sharing the Google note-taking link. It'll be really nice for all of us to be able to have that resource. So thanks to those of you who've already been taking such wonderful notes. And for those people who weren't able to attend all of the sessions, I know they'll be really helpful. In addition, if you are using Twitter, you can use the hashtag reproducibility2020, and that's the same hashtag that you would use if you went directly to slido.com to enter topics in for our unconference session. I think there's a few of you who are having some technical issues showing up in the chat, and we do have technical support here, and they'll reach out and try and help you get those resolved. I would like to, again, thank everyone for coming, and I'd like to thank in advance our next speaker. Ben Margolis is with the Office of Research Integrity with the U.S. Department of Health and Human Services, and he is going to give us a few welcoming remarks. Ben?
All right. Thank you very much, Melissa. I appreciate the introduction, and I just want to thank you for this really wonderful conference so far. Really want to commend you and your team for this really, I think, a spectacular conference, and I know it's gonna continue to be great, notwithstanding whatever job I may or may not do. This is me, and let me just talk a little bit about our mission. Our job is to protect the integrity of public health service sponsored research. The main point, if you see it if you look at our points there, you'll see that ultimately our role is that of oversight. We help promote research integrity through education, but ultimately institutions have primary responsibility. As I think everyone is pretty familiar with our definitions, which the point is to make that it's rather narrow. There's fabrication, there's falsification, there's plagiarism. If we look at what ORI's definition of research misconduct excludes, you see that there's sloppy science, there's bad science, there's even lazy science, which is not within our definition of research misconduct. There's also, as you see in that sort of gray area, some of what we call questionable research practices. It's in a gray area, and irreproducible research is typically a symptom of some or all of those. So the question is, who am I and why am I here? For those of you who might recall this reference, that was Admiral James Stockdale, ran with Ross Perot back in the day. But I just want to tell you more about what we do and about our Office of Research Integrity. This is how we're organized. I'm in this group here, the Division of Education Integrity, Conferences and Workshops, obviously falls within our scope. And here are our marching orders. We foster a research environment that promotes responsible conduct of research. We work to teach the responsible conduct of research, promote research and research integrity. The main point here is there's a lot around education. And so a sampling of some of our activities. One is we're forming a community of practice around virtual RCR education. I'll put my contact information up later for those of you who are interested in getting involved. We also conduct trainings and workshops, which are moving online. We are developing a grant competition focused on data management, which is, I think, very related to this topic. We have active research grant. We fund research grants, and here are some on closely related topics. And of course, conferences like this. One of the things we're best known for are our infographics, as you see, and as well as the other resources you can find on our website. So in terms of advancing responsible research and research integrity, you could call this slide why this conference is important. A big part of advancing science or addressing any sort of issue is naming the issue to help identify the needs. And I think that so far a lot has been done in that regard in this conference, and will continue to be done. It also helps identifying the needs, helps highlight the opportunities to foster responsible research, and needs represent opportunities to support institutions and researchers, particularly on our end. And I have this graphic up here from, I believe it was of Nature. This is to show that really at an institutional level, things don't always work. It's really to say that a lot of it is on us as researchers and scientists to help move things forward. So here are some challenges to responsible research and research integrity. I put in red those that I think have been mentioned to one degree or another. These are all things which help, not help, but lead to some of the problems with reproducibility. So here, I'd like to say, without overemphasizing or not overemphasizing, without being over dramatic, that it really does take a village, that everyone in the research space really has a role, I should say. Everyone plays a role. The quality and the impact of the research that is conducted is everyone's responsibility. Research reproducibility obviously is a vital element of that. All stakeholders must play a role. This is where I would say it's important to emphasize how education fits into this, that it's not just as a student or as a postdoc, but really at every level of experience, this is methods are changing, science is always changing, much the same as we're always learning. We need to continue to evolve and learn. And so just a little bit here, as you, just a little bit to know that obviously this is a topic that is under national academies have addressed. This is something that we fund as an agency. Again, though I should mention, our mandate is actually relatively narrow. We are responsible primarily for the public health service, and that's NIH, CDC, and a few other FDA, a few other agencies, but it's not the entirety of the all the science that is sponsored by the government or that happens. We hope at least to be able to influence those who are our grantees, those who are consumers of the science that is produced by public health service. But where education, in terms of where research reproducibility falls in to this whole grand scheme of things, is that for one, education, and there's education practice, ethical cultures, and it is something that emerge, or I should say, the best practices of reproducible research are a big part of ethical cultures. I say there's a virtuous cycle there. Also, the more that we are able to reproduce, the more we are able to replicate, the more data that we can bring together, the more we can leverage it. There's meta-analyses, a greater power. Research reproducibility strengthens the scientific enterprise in that it helps with credibility. And as we know, credibility is more important than ever, because ultimately science affects the lives of patients. And I'd say everyone at some point will be a patient, at least within the public health service funding scheme. We are funding science that should ultimately affect the patient care and the lives of the population. So this is just a little bit about just a thank you. I just encourage everyone to follow us on Twitter, where we post updates and particularly grant notices and or should say funding opportunity notices. And here's my email. And if you're interested in any way, please contact me. I'm back.
Terrific. Thank you so much for giving us a great introduction, and a special thank you to ORI for funding this conference. It certainly made what we're doing here yesterday and today possible. So thank you for coming, and thank you for all of your help throughout this entire process as well.
All right, thank you. And I'm like I was saying, Melissa, this has really been a great conference, and I look forward to the rest of it as well. The next panel, I think, is going to be a really special one. So I encourage everyone to stick around and enjoy. I think a really amazing pivot. So thank you, and thank you to your team, and thank you to everyone who spent time listening to my slightly propaganda presentation.
Thank you so much. I appreciate it.
Yeah.
Hi, everyone. My name is Angela Abakula, and I'm an outreach scientist at AdTeam. And today I want to talk with you about material sharing and ways we might work together to improve open sharing of materials and reproducibility. So in the life sciences, researchers work with all sorts of materials, like plasmids, antibodies, cell lines. And one major barrier to reproducibility within this field is simply not having access to those materials. And so one part of the solution to this is the existence of repositories like AdTeam that exist to allow researchers quick access to the correct materials that they need that have been deposited by other researchers. And so this is great, but that's only a part of the solution. What the other thing that's needed is widespread adoption of better material sharing practices by researchers. And so at Adgene, we've been working on a few projects to address this. For example, I've been working with journals and publishers to update their material sharing policies to recommend or require deposition of materials with repositories. And we've also been really lucky to be a part of this, to collaborate with the Reproducibility for Everyone workshop group, which is a community-led initiative that empowers individuals to not only learn about best practices in reproducibility in science, but to also run their own workshops. And so these workshops are modular, and one of the modules happens to be about material sharing. And if you're interested in talking with me about material sharing, or to learn more about the Reproducibility for Everyone workshop group, stop by my poster. Thanks.
Thank you so much, Angela. I can't wait to check out your poster a little bit later. We are going to move on to poster B2.
Good morning, good afternoon, and good evening for everyone joining this talk. My name is Shaoqing Chen. This poster is the short introduction about our local community, Taiwan Collaboration for Psychological Scientific Research. You will see how we manage the local open science community in Taiwan. Our country is as large as the Netherlands or state of Maryland, but the population is as much as Australia. However, the public interest to open science and research reproducibility is far, far below these countries. At our beginning, we had the simple idea to build the online study exchange for the junior researchers. We wish our students will learn that psychological studies not only happened in a small room. After the first call for members, we immediately realized our members have to realize the issues that have been discussed in many open science communities. Therefore, we opened the journal club before online study exchange. In this poster, you will see how we run the journal club this year. Later in the virtual building, you will preview the protocol of our online study exchange. Thank you, and see you later.
My name is Ryan Mears, and I'm from the Department of Psychology here at the University of Florida. And I will speed us up in the lightning talk round. I'd like to talk about now and at my poster about replication of psychology experiments, and what we've done in our labs is to give students a chance to work on a replication of an experiment, and the hope is to engage in scientific and statistical reasoning in the classroom. Experiment replication involves a new sample, a novel setting, and yields new knowledge. Replication experiments produce new knowledge for students via enrichment of the linkages between psychological phenomena and theory, as you can see in this diagram here, and to augment experimental knowledge and refine statistical scientific reasoning skills. Yet this approach alleviates any prerequisites of theory mastery and gap finding, which is normally a significant challenge to undergraduate students. So in the end, looking at this map of theory to experiment to phenomenon, because a high fidelity replication will have identical hypotheses and variables that will map from the theory to experiment methods, any differences in what's observed in the experiment will likely come from the environment of the data collection or the new sample itself. This is a simplified problem for students to focus on as they work with the data and analyze the results of their replication experiment. And in the end, they're able to compare and contrast the results of their replication experiment with the results of the original experiment. So in my poster, we'll discuss specific examples of this process and any problems encountered and what we're interested in for future developments. And in the end, this is usually a novel opportunity for students to focus on this particular aspect of doing an experiment because they rarely have the ability to do this in much of their other academic work.
Thank you so much, Ryan. That was great. And to visit Ryan's poster, he will be at poster B3 during the poster session.
Study, which was Intelligibility, Comprehensibility, Accentedness, and L2 Speech by Sarah Kennedy and Pavel Trofimовich. And this slideshow is just a summary of our results, and we hope to go more into detail about what we actually did. So the questions we addressed to answer were how a listener's L2 speech experience and semantic context affects comprehensibility and accentedness of L2 speech. And we did use SoundCloud for the sound files that we did get from the original study, and we used Qualtrics software. And for our methodology, we did have two different groups, one with English speakers with little to no experience with a second language, and one with native English ESL teachers. Both groups were given 3 tasks, which included transcribing in English what they think they heard from 48 sentences, rating those sentences, and a short vocabulary test.
So overall, the results of our replication study were very similar to Kennedy and Trofimovich's original study. There were some slight variations in our data, but none were as compelling to alter our final conclusions. The results of our study seem to strengthen Kennedy and Trofimovich's results, conclusion, and the implications that they make. And once again, the slide on the screen shows our main findings for each of the 3 components of our study that we replicated.
So much, Lindsay and Mackenzie. I will just also add that their poster is going to be poster B5, so make sure that you stop by. And as another note, they did this as part of a class with Dr. Rachel Hayes-Harb. So be sure to attend their poster and ask them some more questions. Thank you so much.
Thanks.
So in the era of the replication crisis, when many classic findings aren't being replicated, the field is essentially looking for models to improve rigor, reproducibility, and transparency. And one method I'm proposing is adhering to clinical practices that are baked into principles are outlined by the organization to help assure the safety, integrity, and quality of clinical trials. Clinical trials are research studies performed in people to evaluate an intervention, and ACTIVE is an example of clinical trial from our lab aimed at investigating the impact of cognitive training on older adults' cognition and everyday functioning. So in a recent paper under review, we sought to demonstrate how single lab studies may model the World Health Organization's principles of good clinical practice for clinical trials using the ACTIVE clinical trial as an example from our lab. So in our poster presentation, we propose that single labs can benefit from using a lab manual, predefined analysis plans, standardized data management protocols, checklists to ensure data is collected in a standardized fashion, the transparent public reporting of all instruments, data, and code, independent statisticians, and quality assurance procedures. So throughout this interactive presentation, I'll provide links to example lab manuals and publicly available data to better illustrate some of my.
Terrific, thank you so much. Brad's poster is going to be poster B6, and we're really excited to stop by and take a look at that and talk with you a little bit more about that.
I'm Elizabeth Wicks. I'm currently a lecturer at the School of Information Sciences at the University of Illinois. I previously worked as a research data specialist at our library. My training is in data curation and data analytics. I focused in reproducibility. I currently teach. So in the past, I've taught workshops on data management and data management plans and all of that, plus data documentation and just and doing consultations with large research groups. I also teach how to program. So I teach Python, R, SQL, OpenRefine, the host of things that are related to reproducibility. And part of my research interest is in how we can help teach people and onboard people into more reproducible work, and using the whole spectrum of reproducible practices. One thing that I've noticed as a challenge in this work is the way that we talk about reproducible practices and the way that we talk about how you learn and work with these things. So in my prior work, I have used behavioral stage theories and behavioral change models and mapped those into more specific data management practices, as far as like my file names and things like that. And trying to use that as a framework to help critically evaluate a workshop series and how we present data management training and things. And as we know, data management and just keeping your business in order is a crucial element towards creating more reproducible work. So I think that there's a lot that can be learned from that process of doing that, and thinking about how reproducible practices is a large network of skills, and that in a lot of ways, when we try to provide best practices to users and to researchers, which is also us too, we're all researchers, we are often doing so in a way that it comes off as a laundry list of either you do all of these things or you're not reproducible, and we imply this binary. And when you sort of imply this binary, if you're not doing the thing, there can be shame and difficulty around that. So I'm proposing that those of us who are working with providing some of these recommendations really think critically about recentering the users in those recommendations. And instead of providing long laundry lists of things that everyone should be doing, we actually think more about, we do need that, right? We need to have the community pushing forward, thinking 10, 20 years ahead, but we also have people who needed to know what they should have been learning 2 years ago. And we have to be able to meet each other in the middle. So I'm proposing that we use more growth mindset vocabulary around how we describe how to learn things, that we provide on-ramps for how to learn these things. So a lot of the talks earlier today talked about early career researchers and how to get people going early so they are better prepared as they go on later. But we still have middle career people and later career people and senior people who are trying to incorporate these things to keep up with stuff. And there's a lot to learn. There's a lot of prerequisite knowledge, especially technical knowledge, to be able to use these practices.
Thank you so much, Elizabeth. That sounds really exciting. For those of you who'd like to learn more, please stop by poster B8. Hi, everyone. Welcome back for just a few concluding remarks. Thank you all so much for attending. This has been a really fun conference, and it's been so great to have discussions with people and learn from all of the different disciplines that are represented. I wanted to let everyone know that we will be sending out information about the videos that we're doing, but we'll also be sending out, more importantly, an evaluation that we would love for you to complete so that we can learn from our procedures and our practices here today. So thank you in advance for please completing that evaluation. Again, I'd like to thank all of you for attending, but I have a number of other thanks to give as well. And so I would again like to thank the Office of Research Integrity for funding this conference. Obviously, without their support, we wouldn't have been able to have the great panel of speakers that we had today, and we are grateful for their support. In addition, I'd like to thank all of the colleagues at the University of Florida who did support this grant, from all of our colleagues in the George Smathers Libraries, the Health Science Center Libraries, our CTSI, and with special thanks to our Senior Vice President, Dr. David Norton, who helped support this and was very encouraging. I'd also like to thank the advisory group of faculty who gave us suggestions at the beginning of who we might invite and gave us other thoughts that helped to drive and shape this conference. And then I'd like to thank our wonderful keynote, Brian Nosek, as well as all of our panelists and presenters. This conference obviously would not have existed without all of the work and support that you put in. So thanks to all of you who were willing to take this on and who submitted content. It was really terrific. But mostly I would like to thank the wonderful team who helped put this presentation and this conference together. And so I'll start with the team from the libraries. And so that's Hannah Norton, Sarah Meyer, Hao Yi, Plato Smith, and Dan Maxwell. Thanks to all of you for all of the work that you did. But with a very, very special thank you to Katie McWilkinson, who was our intrepid conference organizer. And she just did an incredible amount of work to have this conference go as smoothly as it did. So thank you so much, Katie. It was truly, it's been a pleasure working with you, and I look forward to working with you more as we close things out for this conference. The entire conference department at UF was also just incredibly helpful. And so thank you to all of your colleagues who have been helping us out here today, as well as to those who gave us web support as well. So thanks again to the UF conference department and then the planning team. Again, our videos from this conference are going to be up on YouTube. We will be sending links out when those are available. They'll be edited down so that you'll be able to go to whichever video it is that you're looking for. And now I would like to conclude the conference, but by welcoming you, hopefully, to stay for our end of conference virtual mixer. So we're going to take just a very short break, or we can jump directly into our virtual conference mixer. I would encourage you to stay, continue those wonderful conversations that we've been having, and we will hopefully see you there very shortly. Thank you again for attending, and for those of you who helped support or contribute content to the conference, thank you again. I appreciate it. Bye-bye.